let me give you let's see here.
But like, do you understand?
Here's a challenge for you. Okay, do you think conserved DNA sequences actually have any evolutionary significance?
You have it backwards. The fact that something is conserved, something that is significant evolutionarily will be conserved.
Right. So you're saying that conserved DNA sequences must mean that it's functional, correct?
Things that are functional tend to be conserved. It's a correlation. It's not a causation.
Okay. There's actually a study that I read. I can't remember the name of the paper, but I'll cite it down in the comment section, where these scientists, they knock out those conserved sequences, and yet, I think it was in mice. When they knock out those conserved sequences in mice, it actually had no negative effect at all at the mice. How would you explain that?
I would be very, very skeptical, because if you look at developmental biology, when you knock out conserved developmental genes, you have incredible abnormalities in mice.
Yeah, but in this study that I read,
they actually knock
out those conserved sequences, and it had no effect at all.
What conserved sequences? We have so many conserved sequences. If you knock out something like rRNA, the thing that RNA is the ribosome, right? If you knock that out, nothing can live, because it can't synthesize proteins, and that's
right.
Evolutionarily conserved. So you knock out, chromatin packaging, that thing's going to die.
But I think you should read the paper. I'll cite it down in the comment
section.
section.
Vague. What do you mean by conserved sequence?
Now, I know this is a very late response, but anyway, in this debate, Sam and I were arguing in regards to junk DNA and whether or not they are functional. Sam contended that pseudogenes that didn't fall under the transcribed category were non-functional, given that they lacked any practical applications in a biological context. In the debate, however, I questioned him and told him, how do you know they are actually non-functional? What if they are functional but we just haven't detected it yet? In response to my question, he argued that it is meaningless and gave me a rather sarcastic response about him saying how it is theoretically possible for him to bang a chick, but in the end doesn't really mean anything. He then goes on and considers the question I gave him as an argument from ignorance. Now, as a challenge, I asked him, do you think conserved sequences have any evolutionary importance? In his response, he told me it's the other way around, saying that functions in the genome tend to be evolutionary conserved because they are very important for the development and survival of an organism. The point Sam was trying to make here is that DNA sequences that are functional are preserved by natural selection. Sam uses the conservation of sequences as an indication that there is actually functionality going on, a way of determining functional sequences from non-functional sequences. It seems here that the reason why Sam doesn't consider the possibility of other so-called junk DNA regions to be functional is because they haven't been detected. This is the same argument that junk DNA advocates use by suggesting you can knock out those junk sequences out and still have a perfectly healthy organism. Only if you knock out the preserved ones, those conserved by natural selection, would the organism result into abnormalities or cancer. So as a challenge, I told Sam that what if I show you that conserved sequences can be knocked out and have no negative effect in the development of the organism. He was really skeptical about this, and even in the let's just say I'm right scenario, he refused to give me a proper answer. He thinks there is no reason why no negative effect will occur. Regardless, the trick of this challenge was to show that there are functions in the genome that have yet to be understood, undermining the belief that the so-called non-coding DNA regions that appear to have no function are really non-functional. Just because you think a DNA segment is non-functional doesn't mean it isn't. Well, I found a study in regards to mice, which I know I should have brought up a long time ago. The paper is entitled, Deletion of Ultra-Conserved Elements Yields Viable in Mice. Here in this paper, the researchers were interested to see what effect, phenotypically or developmentally speaking, would occur if they knocked out those conserved sequences that they selected. Fortunately, despite myself mentioning highly conserved sequences, these were actually ultra-conserved to the extreme. How ultra was the conservation? The non-coding sequences were found to be so extremely conserved that their base pairs range from 222 to up to 731. In the debate, Sam suggested that perhaps that I have mistaken the conserved sequences as barely conserved, meaning that they were weakly preserved. Here, this is clearly not the case. These ultra-conserved sequences are not the types of conserved DNA segments in general. These are exotic conserved DNA sequences. In the evolutionary paradigm, the more preserved the sequences are, the more importantly functional and crucial they must be to the organism. Using this criteria, one can say that those extreme conserved sequences must be extremely important to the development and survival of mice. Otherwise, as Sam argues, they result into abnormalities, cancer. Here, researchers selected 4 non-coding elements named as UC248, UC329, UC467, and UC482. These non-coding elements showed tissue-specific in vivo enhancer activity in the mouse that tended to recapitulate aspects of the expression pattern found in protein genes that were in their proximity. These protein-coding genes adjacent to the non-coding conserved sequences were evolved in the expression of specific phenotypes. Surprisingly, a deletion and pathological analysis of these 4 ultra-conserved non-coding elements only ranging from each 200 to 700 base pairs resulted in no significant abnormalities nor any birth health defects. Although there were those that resulted into some slightly defects, they were nevertheless deemed as insignificant, meaning that the effect was too weak to be meaningful. Even with an extensive phenotype comparison, the mice that lacked those non-coding regions did not have any distinct phenotypes compared to the wild types. The only abnormalities, defects, and death that did occur were the knockout expression of the protein-coding genes that were adjacent each to the 4 non-coding elements. Nevertheless, the deletion of the 4 extremely conserved non-coding regions revealed no crucial function for the development and survival of the mice. So I asked politely to Sam, why are these 4 ultra-conserved non-coding genes so highly preserved, and yet, when it comes to knocking those sequences out, nothing abnormal or bad happens to the mice? It seems that the assumption that evolutionary conserved sequences is strong evidence for natural selection, since it plays a vital role in survival development, is to some degree questionable, given that not everything that appears to be non-coding or non-functional is really the way they appear to be. My argument against Sam was that because we don't understand everything about genetic mechanisms, it is presumptuous to even state that only conserved sequences play a significant vital role while those that do not are useless. So here's the challenge against Sam. What possible function do these 4 ultra-conserved non-coding DNA regions actually serve when they defy your assumptions about evolution? Thanks for watching.